Good morning or good afternoon, depending upon where you are in the country. It's great to see you all here, and I hope the boot camp is going well for you. Before I start to talk, let me briefly introduce myself. I started off as a hardcore condensed matter theorist, but about 20 years ago, I switched into biophysics. And at that time, it seemed to me that the most natural point of interaction between physics and biology was mechanics. So I got interested in how forces are generated in cells. And in particular, I spent a lot of my career looking at how the growth of actin filaments and their networks generates forces to accomplish different tasks in the cell, like to generate protrusions or to drive endocytosis. Now, before we start in on the measurements of forces, let me first give a big picture of mechanobiology. So to me, mechanobiology is the interaction between force and chemistry. So force influences chemistry. For example, if we pull on a molecule, we know that that will tend to accelerate that molecule is coming apart in most cases. But force can also impact processes going on deep in the cell, like the translation rates of proteins. And it can also impact what type of cell a cell actually becomes when it's mature. And in the other direction, the chemistry of the cell influences force generation. For example, polymers, which can grow, the growth of those polymers is controlled by the concentration of monomers in solution. So it's this feedback loop going back and forth between force and biochemistry that's the heart of mechanobiology. So in today's talk, I'll be talking about measuring forces. But first, I need to say a bit about, in general, about how forces act in cells, because the usual F equals MA analysis is not as useful in cells as it is when we're looking at a baseball, for example. But once we've been through the force balance, we'll then turn to different measurement methods looking at different types of forces, like forces from biopolymer polymerization, forces generated by whole cells and collections of cells, and osmotic or trigger forces, and finally the forces that open channels in the cell membrane. And I encourage you to interrupt at any time to ask a question. If you're lost, I'm sure somebody else is lost as well. The key point to start with is that forces in cells are generally balanced. So let me show you in detail what I mean by that. Here's a cell, and there are various forces acting on it. And in mechanics, we know that the motion or acceleration of an object is driven by the total force, which is the sum of all the individual forces. And it's a vector sum, so let me emphasize that. And that is mass times acceleration. And this turns out to be very small for biological cells. So let me, in fact, work that out. Let's calculate this for a rapidly accelerating cell. So MA, first of all, the mass is the density, which is about the density of water. And then the volume would vary a lot, but maybe 2,000 cubic microns. And then the acceleration, well, a rapidly moving cell moves at about 1/10 of a micron per second, and it might take 10 seconds to change its direction. So that would give an acceleration of 0.01 square microns per second. And that is 2 times 10 to the minus 20 newtons, an astronomically small number, or 2 times 10 to the minus 8 piconewtons. By comparison, these individual forces are on the order of nanonewtons or 1,000 piconewtons or more. So this is way smaller than the magnitudes of the individual forces. And the same holds for the drag force. The drag force is also very small. So we write the drag force as a drag coefficient times the velocity. And notice this is a very different way of thinking about motion. In Newtonian mechanics, we think of acceleration as being proportional to force. And now in cells, it turns out it's really more relevant to think of velocity as being proportional to force. And for the drag coefficient, it's often convenient to think of a sphere, which works pretty well. And if we have a sphere, then the drag coefficient is a prefactor times the viscosity times the radius. The radius, we can say for a cell, the radius might be on the order of 20 microns. And the speed, let's say, again, it's moving quickly, it's 0.1 microns per second. And if we work that out using the viscosity of water, then we obtain a force of 2 times 10 to the minus 13th newtons, or a lot less than a piconewton. So again, very small by comparison with the magnitudes of the individual forces. So the lesson is that all the forces acting on a cell or part of a cell generally balance out and add up to 0. So,
okay. Okay, so now let's go on and work out the implications of that. And let's look at force balance in some particular cellular geometries. So on the left-hand side, we're looking at a cell that has an internal pressure, which would most likely be osmotic. And so you see blue arrows from the internal pressure that push out on the membrane and maybe cell wall of the cell. And on the other hand, the cell wall is pushing back on the membrane, so there is no net force on the membrane. And this is also being helped by the contraction of the cell cortex. If the cell has a cortex, that would be a layer of actin and myosin that tends to contract, and so to oppose expansion. In the context of cells on a substrate, it gets a little more involved. Now there is one force trying to expand the cell. It would like to make it spherical. That's the blue arrows. But there are other components of force that would like to expand the area over which the cell adheres to the substrate, because the adhesion of the cell to the substrate is energetically favorable. And so that actually gives rise to these outward forces that would like the area of attachment of the cell to expand. So all these different forces add up still in such a way that the net force on any particular element, like here, vanishes. Here you can see that there's a sideways force and a downwards force and an upwards diagonal force, and they all add up to 0. So we're basically looking at a situation of close to static equilibrium when we're in a cell. And the same thing happens if we're looking at two cells. In fact, from the point of view of the top cell, the bottom cell looks pretty much like a substrate. And we have those same kinds of forces, some which want to expand the area of contact between the cells, and others which would like to expand the cell and make it spherical. Okay, so when you do calculations in mechanobiology, there's a few simple facts about units which are very useful. First, of course, kT comes up. K is the constant, is Boltzmann's constant that converts temperature to energy. And that's about 4 piconewtons times nanometers, which is, again, a more relevant scale for us. And a megapascal, atmospheric pressure is about 1/10 of a megapascal, or 100,000 pascals. So a megapascal is 10 atmospheres. And it turns out that in mechanobiology units, this works out to about 1 piconewton per square nanometer. So if you had a lot of filaments impinging on a membrane, each of them exerting a force of a piconewton, and there was 1 per square nanometer, they would be exerting a pressure of a megapascal. And it turns out pascals, megapascals, and so on, they're the unit for stress and pressure, and also for elastic moduli, like the Young's modulus. Okay. So if you say the Young's modulus is a megapascal of some, let's say some object, some simple shaped object like a ball or a cube, then that would mean that if you exerted a megapascal of force density on that object, it would deform really noticeably. It would deform like by a factor of 2. So, anybody want to hazard a guess at the Young's modulus of Jello? Okay, just raise your hand or unmute yourself if you have any idea what the Young's modulus of Jello might be.
Like 10 kilopascals?
Well, that's a decent ballpark. I would have guessed 1,000, but I'm curious. What do you base your guess on?
I've made hydrogels in the lab that are about 10 kilopascals, and when I squish them, they kind of feel like Jello.
Oh, yeah, fantastic. Well, that's a much better answer than mine then, because I've never made hydrogels in the lab. Yeah, I'd say Jello is somewhere in the range of 1,000 to 10,000. And the reason I mention that is that the constituents of cells often have a Young's modulus of that general magnitude. Like the actin cortex will often have a Young's modulus of 1,000 to 10,000 pascals. So it's kind of like Jello. So in many ways, the inside of the cell is like Jello. Okay, now, so the first topic we're going to look at is measuring polymerization forces. And so, for example, a polymerizing actin filament or a polymerizing microtubule. But to do that, we usually look at the shape of the filament itself, because that gives us the force scale. So you need some kind of, we're extracting data from micrographs, and so you need some force scale to figure out what a certain bending means, for example. And an important property is the stiffness of a biological filament, which is quantified by its persistence length. And so to get the idea of what a persistence length is, imagine that we have a filament in a cell and it's undergoing thermal bending from interacting with molecules in the solution. And then the persistence length, imagine that you were riding like a roller coaster and you felt your direction changing over time. Well, the characteristic distance over which your direction changes would be the persistence length. So now the filament is your roller coaster and you're riding along it. And as you change direction, then the distance over which that happens is called the persistence length. And there's another more direct measure that has units of energy in it, but that's a little harder to remember. The persistence length is a much easier one to remember. And now, so what I'm really talking about, think of this ruler as a filament. I'm going to have to step back a little bit here. Okay. So I push on it and then you can see that I bend it. Okay, so that's what we mean by the stiffness. It's resistance to bending. Okay, and anybody want to hazard a guess at the persistence length of this ruler?
$20 meters.
Okay, I got a guess of 20 meters. Any other guesses?
Half the length of the ruler.
See now, we're coming up against something crucial, okay, and it's in the fine print. If you look back at the PowerPoint slide, we're looking at a thermally bending filament. Okay, so here we got me. Well, at least compared to thermal forces, I'm big and strong, and so I'm able to bend this thing a lot. But now imagine if it were only being bent by thermal forces. How long would it take before it completely lost memory of its orientation? So for that reason, certainly I'm holding it here, and you can see it's pretty stiff. So the persistence length has to be a lot greater than the length of the ruler itself, because it's not bending thermally at all, really. Any bending is either coming from me or gravity. And the bending in the persistence length is all thermal. So given that, do I have any other guesses about the persistence length of the ruler? Well, okay, it's something to give some thought to, and it turns out the answer is it's about the size of the universe, actually. And turns out that even a stick of spaghetti has a persistence length that's really huge. And so what that tells us is that thermal energies are small on our scale, but on the much smaller scale of a biological filament, they can become very important. Okay, so this definition of the persistence length gives us a way that we can measure the persistence length. And the fact is, this comes from thermodynamics, that the square of the magnitude of the fluctuations is proportional to temperature on the one hand, and it's inversely proportional to the persistence length. And so that means essentially that if we quantify these fluctuations in some way, and I'm not going to give the math on how to do that, then we will have a good handle on the persistence length. And so let's watch it. There we go. So there's a filament fluctuating thermally. Turns out it's a couple of actin filaments that are glued together. You see there's a piece in the middle that's a little brighter, and that's where there are two filaments. And you can, in fact, see that part of the filament is bending less than the other part because it's stiffer. There are two filaments at that point. And so what they do is they take these images and then they analyze how much the filament is bending, and they assume that it's all due to thermal bending. There's certainly nobody reaching in there and bending it by hand. And in that way, you can extract the persistence lengths of various biopolymers. So here's a nice collection of data. So for DNA, it's very short, because we know that DNA is very tightly packed in the cell. You couldn't do that if it were too stiff. For actin, it's about 10 microns. So that means it's a little smaller than the size of most cells. And for microtubules, it's huge by comparison. It's 2 millimeters. Okay? So a microtubule, if you left to its own devices on the scale of a cell, would be basically completely stiff. We see microtubules bend in cells, but that's because they're getting forces exerted on them by molecular motors. So let's turn now to polymerization forces. What's happening here is that a biopolymer is polymerizing by adding a subunit at a time, so going from here to there. And this is an exothermic process. That means the free energy change is negative, and the maximum force that can be generated is the magnitude of the free energy change divided by the step size. So basically, each one of these squares here is 2.7 nanometers. That's the step size for an actin filament. And so this is a prediction that it should be possible to generate about 3 piconewtons of force by a growing actin filament. And this depends on the actin concentration. That's where the free energy has to do with the actin concentration also. So I just chose a particular value here, but that's the general ballpark that the force scale is on the order of piconewtons for forces generated by biopolymers. Okay. And so here's a simulation that I ran quite a while ago where this process is being illustrated. So we have an actin filament that's trying to grow against the membrane, and this membrane has a downwards force on it in this coordinate system. And it also is experiencing random Brownian motion. And so I simply ran this simulation until the subunit got added. Okay. And so here we go, that subunit, there's one at the end, it's trying to get in and not quite, there it is. There it makes it in. Okay. And so this is what happens. These subunits add one after the other. And the kind of thing that you like to look at in these simulations is the force-velocity relation. You want to know what kind of motor is this. A really strong motor is one that can exert a lot of force and run fast at the same time, because power is force times velocity. And so there are many different kinds of biological motors. This one we predicted, and people before this had predicted with simpler theories, that the velocity of polymerization would decay very rapidly, exponentially, in fact, with opposing force. And that's what we found in the simulations. We found that the actual results were somewhat below those of the simple theories. But the idea is that when you exert an opposing force, that really slows the polymerization process. Okay, so this is one of the very few measurements of force generation by polymerization of an individual biopolymer. So this here, this is a little submicron piece of an actin filament, and it's anchored at its two ends in different ways. At one of its ends, it's anchored by a dead myosin, and it just sits there. That end doesn't do anything; it just sits on the substrate. But at the other end, it's anchored by what's known as a formin. And formin is a molecule that can anchor the actin filament on the one hand, but the filament can keep polymerizing while that formin is attached. So even though this filament is attached to the substrate, it can continue to polymerize and generate force. And so the question, or excuse me, the experiment worked by seeing if the filament could generate enough force to buckle itself. So there's a mechanics calculation for the buckling force, and you don't need to worry about the details here. The point is, this is something pretty well known from classical mechanics. And for a filament of this length, 0.7 microns, that buckling force would be close to a piconewton. So in other words, if the filament can buckle itself, then we know that it's exerting at least a piconewton of force. So here we go, let's watch it. Yep, there it goes, it buckles. Okay, that was really fast. So let's watch it again. Okay, so that filament is able to buckle itself. So that's not very detailed information, but it does tell us that filaments consistent with the theory can generate forces on the order of piconewtons. And here are some more detailed studies of microtubules. Microtubules are easier to study just because they're stiffer. And so you can get a microtubule of a length of several microns and still have it generate force. Whereas an actin filament that's several microns long will not be able to generate any appreciable force. And in this experiment, what's happening is that there's a glass slide here, and there's a specially shaped well. The microtubule is put in that well, and it's glued to the bottom of the well here, and then this other end is free to grow. And then they look at it from the top, and they get pictures like that. And especially if you go down here, you see here's your straight microtubule, and now it hits the edge there. This thing has gone over and hit there, hits the edge, begins to bend, and then it bends farther and farther. And because we know mechanics, we're able to figure out how much force is required to bend the microtubule. But we can also look at the total length of this microtubule as a function of time, so that gives us the velocity. And this is the result that, in fact, yes, the velocity does decay exponentially with the opposing force, but it's actually off from the theory by about a factor of 2. The theoretical curve would have gone up here. I guess they were too embarrassed to show that in the paper, but if they had, they would have showed that, yes, we kind of confirmed the theory, but not quite. A very common situation. Okay, and now you can do this, work your way up and look at progressively larger numbers of filaments. So here is an experiment where this whole entity here, it's called an acrosome, and it's got a bead attached to it. Filaments get grown out of that entity. And the acrosome is held in place in a special kind of trap that also freezes its orientation. So it's almost like you can hold this thing here by hand and then have it grow against an obstacle, which is just a ledge on the slide here. And then it's held in an optical trap, which lets you measure the force. And then at the same time, you can measure the number of subunits added. And then you find these kind of plots of the force, if you look on the right-hand side, is a function of time. And these are different actin concentrations. But what you're seeing is that the force is on the order of a piconewton, which actually surprised everybody because there are like 6 to 8 filaments that are exerting a force. And so if they're each exerting a piconewton or more, we would have expected forces on the order of 5 to 10 piconewtons. So this was actually kind of small. And that was kind of a surprise. And we still don't quite understand the reason why this occurs. And similarly, this is now an even larger collection of actin filaments. There's a bead here that nucleates actin filaments, and this is the comet tail. And this whole entity of the comet, the so-called comet tail attached to the bead, is attached to a cantilever here, whose stiffness we know. And then it's attached to a pipette here, which allows the experimenters to exert a pulling force on the comet tail. So they can exert both pulling and pushing forces on this growing actin comet tail that's nucleated off the bead. And so here, this is what you see. It's not really that detailed, but they were able to extract the data from it. And they find that the force, it decays, the velocity does decay with opposing force, but it doesn't seem to be decaying that rapidly. It really seems to be at a long force tail. And one reason for that may be something we know now, which is that when you exert force on an actin network, you enhance the nucleation of new filaments. So there may actually be more filaments at this end than there were at that end. And that is enhancing the force generation capability of this system. Okay, so let's move on now. We've been looking at actin filaments and collection of actin filaments. Let's now look at a whole cell. This is a little bit easier because we can use a cantilever that's familiar to us. We can calibrate it and so on. And the experiment is simply that we have this cell growing against the cantilever, pushing on it, and then we measure the orientation by a mirror that's sitting up here. So there's a light ray coming in and out. And then you're able to measure the motion of the end here and the force as a function of time. Okay, so here's what happens. before we have plenty of time, so let me show you what we got. So this is a cantilever here. It's got that diamond-shaped thing at the end, and here's the moving cell. It's a keratocyte, and keratocytes are incredibly steadily moving cells. They come from fish scales, and a lot of other cells change their mind a lot, but they're very single-minded cells. They just keep going in the same direction. Now watch closely. Soon something's going to happen. You've seen the cantilever has been slowly moving a bit to the right, and in just a second it's going to pop back. So what happened is that first of all it hit a lamellum, and then it contacted the nuclear mound, and then maximum force, and now the cell breaks through. Okay, so you can literally see the forces being exerted by the cell as it went through there. And so this is a quantification of that. So it turns out the most interesting thing for us is this first part here, and that's blown up over here. And the cell hit the lamellipodium of the cell, first makes contact here, and then the force starts to climb. And it turns out that then a funny thing happens. It's as if the lamellipodium gets wise and it starts to sneak around. So it sneaks around the cantilever, and then the force drops while it does that. The other parts of it that were on either side of the cantilever move ahead, and then it drops down, but then at some point it hits the mound of the cell, and then there's no stopping it. But the bottom line is that the total force is on the order of nanonewtons, which makes sense. We talked about piconewton forces for individual filaments, and now we have nanonewton forces for the whole cell. Okay, right. So now we're going to ask for something a little bit more detailed. Okay? We're not just going to talk about the whole force, but we're going to talk about the distribution of force. Okay, so here is a cell with a nucleus and an actin network. It attaches to the substrate the focal adhesions, and then we'd like to measure the forces exerted by those focal adhesions. And one way of doing that is to use fluorescent beads. So these are embedded in an elastic material. Okay? And so you can use gels of varying stiffnesses. So here the beads are like 300 to 700 nanometers in diameter. And then you take a picture of the substrate without the cell, and the beads are in a certain position. And then you put the cell down, and it exerts forces on the substrate, and that moves the beads. And then from those displacements of the beads, you can figure out how much force was exerted in a given region of the material. And so you can imagine that's tremendously complicated math. And that's actually been one of the things that's been limiting the development of this method. It's pretty much taken care of in 2 dimensions, but in 3 dimensions, you can do the same thing. And then the math really gets elaborate, and its computer efficiency really is, even though computers are so fast, the computations are still pretty challenging. Okay, so here is an experiment where cardiac myocytes were put on a substrate. And this is the cell here. And here these observation points are 5 microns apart. And so they put the cell down, and then there are all these fluorescent dots that you're seeing in the matrix. And they measure the motions of those dots. And that's what's shown here on the right. The yellow is the displacements, and the red is the forces. So it's a little hard to make it out, but in fact, the forces are all inwards. So you put this cell down on the substrate, and it automatically starts contracting and pulling in on the substrate. So the cell is a very active entity in this case. What I found really surprising is that they added up the sum of all the contractile forces. Now they didn't add them as vectors. If they had added them as vectors, they would have all added up to 0. But they just added their magnitudes. And so what they find is that oscillates over time with a period of roughly a second, which sounds kind of familiar, doesn't it? I mean, that's like a heartbeat. So it's saying that this little myocyte by itself actually seems to know enough to exert a force that varies on the same timescale as a heartbeat. So these cells are pretty smart guys sometimes. Something else that was really interesting is that cells can measure the stiffness of their substrate. And so what you're seeing here is, that as a function of the Young's modulus of the substrate, again, the sum of the contractile forces all of a sudden starts to take off when the Young's modulus gets to be like, 50 to 100 kilopascals. So the cells are able to measure the stiffness of their environment. And that's important because cells have to know where they are. And that's one of the features of where they are and where they should go is the stiffness of their environment. Now here's another way of doing this that's a little bit cleaner. It's harder to set up. You have to really have like a nanofabrication laboratory. But you can use micropillars to measure the forces. And so here you have an array of micropillars on a substrate, and then the cell settles down on those micropillars and pulls them in, and the chips move in. Okay, so it's the same kind of thing. It's just that the analysis is much simpler because the posts are independent of each other. So you can look for, at any given post, the displacement of that post is equal to the force that's exerted on it. So you don't need to do this very complicated elasticity math. You can just look at the displacements of the individual posts. And that's what you're seeing here. You see that up here, the posts are pointing pretty much straight up. But now around the cell, they are being bent toward the middle a little bit. And it's the extent of this bending that you measure. And here they found each post had a force in the several nanonewtons or more. And they found that the force per post increased with the spreading of the cell. And it's not known completely why. The natural thing would have been to say that, yeah, a spread cell exerts a larger force than an unspread cell. But this is saying even more. This is saying that it exerts a larger force per post. So it's a phenomenon that there's no definite explanation of that. Okay, and we can also look at layers of cells. So now we look not just at a single cell, but at like something like a tissue. It's not a 3D tissue, but a layer of cells. And then we had to think in terms of stress because it's getting to be a macroscopic entity. So let's step back a little bit. Now here, if you look on top, so these are our cells, and you'll see that they're kind of sparse here, and they're more tightly packed, sparse, more tightly packed, and so on. Okay, so it turns out that something that's under positive pressure or compressed has a negative stress. So that's why we have a negative stress here. And over here, we have a positive stress. And now the question is, how is the stress going to be related to the force density? And what happens is that because this is dense here, and this is not so dense here, if there's a boundary between a region of higher pressure and lower pressure, then there will be a force that goes from that region of higher pressure to the region of lower pressure, which here would be in the negative direction. So here is where the rate of change is the most rapid, and what we see here is that the force is negative there. And so we can look at both the stress and the force densities. So here is a layer of cells with a boundary here. And this was, again, done using fluorescent beads. Here you can see that the beads, let's see, the green ones are the ones before the cell was put down, and the red ones, sorry, backwards. The red ones are the ones with no force, and the green ones are the ones with force. And you can see that here, red going to green, and here's some overlap. You can see that there is a displacement. You can also see that it might be hard to measure it that accurately, and that's the case with these systems. And so here is the distribution of traction forces. And it looks kind of discontinuous. It's hard to make that much out of it. And so it's easier to look at the stress. And so what you're seeing here is that there are foci of tensile stress. The highest is like about 400 pascals. So that's about the jello again. And then there are other more broadly distributed regions of tensile stress, or excuse me, compressile stress. Okay, now let's step back and go to a completely different world. Let's look at molecules. Okay, it would be great to be able to measure the forces at a molecular level inside cells. That's really important because otherwise we don't really know how the forces are impacting the biochemistry. And so this is something that's been developed very rapidly over the past 10 to 20 years. And here's an example of a particular type of molecular tension sensor. So here is the inside of a cell, and here's a focal adhesion. And that cell is on a substrate, which is down here. Here's an integrin, and the integrin would ordinarily link to the substrate. And instead, they made this construct where it links to the substrate via an elastic entity that can stretch or be unstretched. So you have both these possibilities. And now, so the length of the spring will be determined by the tension. If the tension is high, it'll be stretched. If it's low, then it'll not be stretched. The readout for that is the fact that when these two molecular entities, one called the donor and the other called the acceptor, when they are close to each other, that limits the fluorescence from the donor. So ordinarily, the donor can go off green fluorescence. But when the donor is close to the acceptor, then that is much less efficient. So if you see the fluorescence, that means the force is low. If you don't see it, it means the force is high. And so here's the efficiency, and you can see that with increasing force, then the fluorescence remains, which means that the fret is not occurring. And fret, we just call it fret. It means first or resonance energy transfer, but doesn't say that much for both of us. The point is that the fret is the quenching of this fluorescence by these molecular entities being close to each other. So what you can do is measure the fluorescence, and you can see, are you up here or are you rather down here? And that'll tell you something about the force. And so here's a result of a measurement like that. They're looking at paxillin, which is connected with focal adhesions. And so they're plotting, on the one hand, the paxillin distribution on the substrate, and they're also plotting the tension. And they're seeing that there's a very close correlation between the tension and the paxillin. So in other words, where the cell is exerting a strong force on the substrate, that's where the paxillin is. So it tells us that the penicillin is really involved in exerting the force on the substrate. And finally, this is not quite part of the story, but I just think it's really neat that you can unfold a single protein by exerting a force on it. So you can grab either end of a protein and then start pulling on it, and as a function of distance, you unfold domain after domain after domain of this protein. And the force scale for that is only over 200 piconewtons. So that takes a lot. Okay, another way we can measure stress directly is to use laser ablation. So look at this filament here, and now step to the right. Notice that a gap is developed, and this because a laser beam blasted that fiber and broke it. Okay, so what you see now is it initially starts to pull apart, and that means that that fiber was under tension originally. Okay, if it had been under compression, it would have gone the other way. So we learned something from this, that it was originally under tension, and then it keeps moving farther and farther apart. And so it takes some mechanical modeling, but from this, we can determine the final displacement, and also some things, if we know the viscosity of the medium, we can also get some idea of what that initial tensile stress was. Okay, now let's, before we look at this, let's remind ourselves of a little bit about osmotic pressure. Okay. So let's see, there, okay. So let's draw ourselves a cell with a cell wall. Okay, and then we're also going to have a membrane inside that cell wall. It's supposed to actually be touching the cell wall, and so I'm almost managing that. Okay, and now, oops, let's have a different color there. There, okay. All right, so we're going to put a bunch of molecules in here, and then we're going to put fewer molecules here. Sorry, those molecules came out as lines, but I think you get the idea that we have more molecules inside the cell than on the outside. And then the fact is that it's almost like an ideal gas, where we have we can calculate the ideal gas pressure from the solute molecules inside the cell relative to the outside of the cell, and we get that the osmotic pressure, which we call pi, is equal to kT times the inside concentration minus the outside concentration. Okay, now, so that's fine, but let's, how would we measure this osmotic pressure? It's actually hard to get it. If the cell is really big, you can get a sensor in there, but otherwise we have to do something indirect. Okay, and so here's one way of doing it. So here's a soccer ball. Okay? And the soccer ball, think of the leather as being the cell wall, and then the kind of the rubber thing on the inside is the membrane. Okay, now if I wanted to measure the pressure inside that soccer ball, what I would do is squeeze on it like that. Okay, I'm not doing a great job here, but this is about all I can manage. So, and what happened is that once I exerted a pressure that exceeds the pressure on the inside, then I will be able to bend it, so or be able to buckle it. So that's how our methods of our standard methods of measuring the osmotic pressure work. so what we do is we increase the force that's called the external osmotic shock. So we increase the concentration of osmolytes on the outside until there is kind of a rapid collapse. And that's what we see here. So you follow the volume in the experiments as a function of the external concentration, that all of a sudden it'll have a cusp and it'll dip down. And that's the point where you've added just enough to counter the initial excess on the inside. And here's some actual data for that. So this is for yeast, and here's the curve, and you find that if you go down to that point, that gives an osmotic pressure of almost a megapascal. Now, the trouble is that when we push in on that yeast cell, it pushes back, and it starts generating more osmolytes. And so that's as if, in the case of the soccer ball, when I'm pushing in on it, somebody else decides to start inflating the soccer ball at the same time as I'm pushing in on it. And that's going to make it very hard for me to make it collapse. And I'm going to underestimate, or I'm going to overestimate the pressure in the soccer ball. And the same thing happens in this case, that in fact, this is probably a very large overestimate. And so a more accurate method has been developed lately on the basis of this physics experiment involving a real rubber ball, where they found that they were able to measure the pressure inside this rubber ball by exerting a force with kind of a poker like that. And they found, using both experiment and theory, that the spring constant of this poker, once you got it in far enough, it became constant, and it was determined by the pressure inside the ball. So in other words, if you could do this to yeast, if you poke it just right, and measure that spring constant from the poking, then that'll tell you the osmotic pressure in the yeast. And so here's that kind of experiment. You measure the spring constant. At first, it starts off kind of small, but then it reaches an asymptotic value as a function of the amount of indentation. And the result is that now you get 0.2 megapascals instead of 0.9. So that's a big difference. And I think this is the more accurate value in this case. The reason this experiment is better is that it's done in a very rapid time scale. It's like a millisecond. And cells take like minutes to respond. And so the cell simply does not have time to respond in this case, and you're measuring the real osmotic pressure.
Hi, Anders. This is Patience just checking in with a 4-minute warning.
Okay, very good. Yep. And just want to show one more thing, that there is the mechanosensitive channels are an important area of study in the group. And so it's important to be able to measure the tension at which they open. So here's a channel, and now the picture is that as the tension in the membrane is increased, that channel will open and let osmolytes in. And that actually can be changed via the pressure in the cell. And so the way we measure that is to put a suction pipette on the cell. And then if you look here, here's a little boundary. This is the boundary of the cell. You can see that when you pull on it, that then this bends. And by evaluating the change in pressure at the same time as you pull on it, you can work out the tension in the membrane. And then you can measure the current through the mechanosensitive channels. And so you can get the opening pressure of the channels. And that's what you're seeing here. This is for a particular bacterial channel. You'll see this is the open probability, and it's very small until the tension is here like 6 times per centimeter, and then it levels out. So I'm mentioning this really because it's an important topic of study in the group. So that's it. It's been, I guess I tried to sound optimistic as I gave the talk, but in fact, we can still only measure a very limited range of forces in cells, and we're far from being able to get a map of stress throughout the cell. But new technologies are pushing the field forward, and this is definitely something worth devoting your efforts to if you feel like it. So I'll stop there, and I'll be happy to take any questions.
And we definitely have a couple minutes for some questions, just a couple minutes.
I have a question.
Yeah.
So my question is about using micropillars to measure forces.
Yes.
Because I'm thinking with the pillars, the cells are elevated, and then their contact area, like the contact area between the cells and the pillars, are less or is less than if the cells were on a flat matrix. So
Yeah.
how does that change or affect the forces that the cells are generating? And also, how does that change how they spread?
Right. So the fact is that any surface that you put a cell on is going to be kind of artificial, because in a tissue, the cell is surrounded by the ECM, typically a fibrous ECM. And so we now, when we do most experiments, we just put a cell on a substrate, and that is one maybe not so great approximation. We put them on the pillars. That's another approximation, which may also not be that great. But to me, they're just two kind of different approximations, and I think the strategy is that one would want to look at both kinds of experiments. Because anything that comes out of one type of experiment could be an artifact, but if you observe the same phenomenon coming out of both types of experiments, that would give you a lot more confidence. Also, what happens, usually the cells exerted by the force on the substrate are not uniformly spread out over the substrate. They occur at certain adhesion points. And so probably what happens in the case of the micropillars is that those adhesion points simply get focused on the micropillars. And so in that sense, it may not make that big a difference. But the take-home message is that any experiment done on just a purely two-dimensional experiment, you really have to think about how relevant is this for a true three-dimensional cell.
Thank you.
Sure.
So my question is, like, when you talk about osmolytes, what kind of molecules are you talking about?
Yes, right. Let's go to that picture. Oh, I think that was actually, let me pull up the whiteboard. Right, okay, right. So these red guys here, what are they? Well, they can be many different things. They can be ions. For example, they can be sodium, potassium, but they can be many other things also. Like in the case of yeast, they can be glycerol. And so what's really amazing to me about the osmotic pressure is that it doesn't necessarily matter that much what kind of molecule it is inside. That's the crazy thing. What really matters is that you have a setup where we have molecules on the inside that can't easily move from the inside to the outside. They're kind of trapped. But the water is not trapped. The water flows back and forth easily across the cell wall and the membrane. And so, you can really think of different limits. So you could think of one limit where the water does not interact with the osmolytes at all. Okay, then the water is completely irrelevant. And so then you would just see the pressure of this gas of osmolytes on the inside relative to that other gas of osmolytes on the outside. And then we know if the osmolytes are denser on the inside, they will be exerting a force that pushes it out. You can also think about it in terms of if the waters have an attractive interaction with the osmolytes. In that case, the water will actually be sucked in to the cell if there's a larger osmolyte concentration in the cell. And that would be another mechanism that the pressure is enhanced. And one thing that I find miraculous is that the final result is pretty much the same in either case, in both of those limits.
Okay, so I guess my greater doubt was that I know that in the nucleus there's a lot of charge balancing going on with the DNA because it's mostly negative and chromatin has like different charges. So it's shuttling in and out a lot of ions, but then this is also a mechanism that the cell uses to control the overall pressure.
Yes.
So then would the pressure that's on the cell be correlated with the like manipulation of the DNA, I guess?
Oh, I see. Right. Okay. I think the interaction between those two effects is not that large. And the reason is that the DNA has it's got a high charge density, but inside the cell there are free ions floating around, and so those will tend to neutralize that charge density. So there's charge density of the DNA in the nucleus, but we don't really see that farther out.
Okay.
So it ends up being that just what's the overall density of ions in in the fluid that's coming into contact with the membrane. Thanks.
I had a question. So going back to focal adhesion sites, if a matrix is it correlated that matrix stiffness correlates with a number of focal adhesion sites? And if so, could a cell confuse a soft matrix with high ligand density with a stiff matrix with lower ligand density?
Okay, let me go back and first pull up that picture again. So let's see. Right, you were thinking like here, for example, in the adhesions. Right. Okay. So now you were wondering to what extent the density of adhesions depends on the mechanical properties of the substrate.
Yes.
Okay, yeah. Let's see. Nothing is actually coming to mind directly on that, but my expectation would be that they would be increased because you need more focal adhesions to be able to exert a large force. So my guess would be that it would enhance the growth of the focal adhesions.
Okay.
Any other questions before we sign off for lunch? And Anders, would you take email questions if people come up with?
Yes, I'm very happy. Yeah, go ahead, email me. Yep, please do.
Okay. And we will upload Anders' slides to the box as well. And the